Good evening, class.
Good evening, sir.
Yes, yes, sir.
All right. Tonight we are going to talk we want to talk on bacteria metabolism and fluorescent antibody technique. And then let's start without wasting time. In bacterial metabolism, we need to talk about the energy, matter, and enzyme. We talk about catabolism of carbohydrates, catabolism of lipids and protein. Then talk about Krebs cycle, otherwise known as TCA tricarboxylase cycle. And then we'll touch some other things that's at least for our knowledge and information. Cellular processes such as building or breaking down of complex molecules occur through series of stepwise interconnected chemical reactions called metabolic pathways. So we have a series of them and they are interconnected. They are chemical reaction called metabolic pathways. And the term anabolism refers to those endogenic metabolic pathways involved in biosynthesis, converting simple molecule building blocks into more complex molecule and fueled by use of cellular energy. What this one means is that once you talk about anabolism, you are talking about biosynthesis, where simple molecules form a building block to become complex molecule. So that is anabolism. But catabolism is like you are breaking down the complex molecules to smaller molecules. So that is the difference between anabolism and catabolism. And then we have what is called heterotrophic metabolism. So this one involves biologic oxidation of organic compound. And what are those compounds? The compound include glucose and to yield ATP. And what is ATP? Is energy. So, and simpler organic compound which are needed for bacterial cell for biosynthetic or assimilatory reaction. We all need, whether man or microorganism, we all need glucose to generate ATP, which is energy. And then let's go to catabolism of carbohydrates. In the catabolism of carbohydrate, the first step there is glycolysis, so it's breakdown of glucose resulting in the formation of ATP, which is produced by substrate level phosphorylation. We have NADH, nicotinamide adenine dinucleotide. The reduced form is NADH, and then we have two pyruvate molecules. Glycolysis, as we have read before in our biochemistry in 200 level, does not involve the use of oxygen. So it's not oxygen dependent. So after glycolysis, we have 3-carbon pyruvate that is decarboxylated to form 2-carbon acetyl group. Coupled with this NADH. I've told you the full meaning of NADH. NADH is a reduced form of nicotinamide adenine dinucleotide. And then the acetyl group is attached to larger carrier compound called coenzyme. That is for catabolism of carbohydrate. It's more complex than this, but we just need to get something out so that we will not if you want to do catabolism of carbohydrates, we can use the entire lecture for that. So another one is respiration. which is a type of metabolism that uses oxygen. You know, we are talking about glycolysis that is not oxygen dependent. Respiration is oxygen dependent, and in which 38 moles of ATP are derived from oxidation of one molecule of glucose and yielding 380,000 calories. Respiration is energy producing process, and the cells, whether human cell or microorganism cell, depend on this in order to survive. Mhm. So we have fermentation. This is another heterotrophic metabolism. There is an organic compound that is involved that is not oxygen. The organic compound is called hydrogen, and then what this hydrogen does is to accept electron in the process. So um, I told you about NAD plus, NADH rather. Then we have in fermentation, there's one organic molecule that serves as final electron acceptor to regenerate NAD. NAD means oxidized form of nicotinamide adenine dinucleotide, NAD plus. But from NADH, the NADH is the reduced form of nicotinamide adenine dinucleotide, which carry high energy electron and a proton. You know, we are talking about that fermentation does not use oxygen, but it uses a hydrogen, which is a proton known as hydrogen. So the primary role of fermentation is to recycle the NADH back to NAD plus, so that glycolysis can continue to produce small amount of ATP in the absence of oxygen. So respiration depends on oxygen, while fermentation does not depend on oxygen. So microbial fermentation have been used for production of foods and pharmaceuticals, and for identification of microbes. So there is another one that we call the catabolism of lipids and protein. Collectively, microbes have the ability to degrade wide range of carbon source beside carbohydrates. We know in our elementary biology, once there's a process called gluconeogenesis. Even in biochemistry 200 level, I think we are taught gluconeogenesis. When there is no, or the carbohydrates is in short supply, the body will find a way of getting its energy through lipids and protein. So that's what they call um, gluconeogenesis. And then in the catabolic pathway for this, all these are connected together. The glycolytic pathway is connected into the Krebs cycle, that is tricarboxylic cycle, and then all these things, they are they work in concert. One cannot work in isolation. They have a way, they have a meeting point where one leads to another. So several types of lipids can be microbially degraded. So when you say catabolism the other time, it's breakdown of a complex molecule to smaller molecules. So when you have lipid, the lipid will break down into diglyceride, and then thereby releasing fatty acid from the glycerol backbone. Then phospholipids are also degraded by phospholipases. That's an enzyme releasing fatty acid and phosphorylated head groups. So I hope this one is just a refresher course for us, because all these things we are taught in 200 level biochemistry, and then so that's why we are not really going deep into it, but just for us to refresh our memory that there's something like that, and the microorganism make use of them. I mentioned the Krebs cycle the other time, that is otherwise called carboxylic acid cycle or citric acid cycle. This one is oxidative process and respiration by which pyruvate is completely decarboxylated to carbon dioxide. This pathway yields 15 moles of ATP, unlike respiration that yielded 38 moles of ATP. We also have glyoxylate cycle. This also occurs in bacteria and it's a modification of Krebs cycle. And we have bacteria photosynthesis, just like in plants. That's what they call bacteria photosynthesis. So the bacterial photosynthesis is light dependent, anaerobic mode of metabolism. Is anaerobic, that is not oxygen dependent. Carbon dioxide is reduced to glucose, which is used for biosynthesis and energy production. So bacterial photosynthesis is also very important. Although photosynthesis is most commonly associated with plants, microbial photosynthesis is also a significant supplier of chemical energy. So they supply chemical energy to microorganisms, fueling many diverse ecosystems. So we have autotrophy. Autotrophy is a unique form of metabolism found only in bacteria. Inorganic compounds are oxidized directly without using sunlight to yield energy. The other one uses sunlight. This one does not use any sunlight. And then they require energy for carbon dioxide reduction. Another one is anaerobic respiration. This is also another heterotrophic mode of metabolism in which specific compound other than oxygen serves as terminal electron acceptor. So we have nitrite, we have sulfates, and even carbon dioxide for methane producing bacteria. Then nitrogen cycle. We were taught this nitrogen cycle in our secondary school agriculture. We have ammonification in the air, nitrification, denitrification, and nitrogen fixation. These are primarily carried out by bacteria. There's another one under this bacterial metabolism is called biogeochemical cycles. As energy flows directly through ecosystem, entering as sunlight for phototrophs or as inorganic molecule for chemoautotrophs. So we have all these ones. So this one, let us move to some method that use biochemical characteristics to identify microorganisms. We have some organisms that they use observable traits and structural components such as lipid, sugar, or amino acid, and they store compounds such as poly B hydroxybutyrate. The example of these are the non-fluorescent pseudomonas. The fluorescent pseudomonas are Pseudomonas aeruginosa. I hope you know it. Other form of pseudomonas, they can use this method to identify them. Example of it is we have the fluorescent one that I was telling you the other time, which is Pseudomonas aeruginosa. But this method is used to identify the non-fluorescent Pseudomonas. And then we have another one that is called MADITOF. And it's an acronym that is known as matrix-assisted laser desorption ionization time of flight. So that is the technique, and it's an automated system that identifies microorganisms. And this one determines mass spectrum and then comparing to the database. There is a database of a known organism. So whatever we get here, we now compare with the database, and we are able to identify our organism. Another one is the determination of lipid composition using fatty acid methyl ester, FAME, fatty acid methyl ester, or phospholipid derived fatty acid PLFA analysis. So this also can be used to identify microorganisms by measuring their unique lipid profile. This one measures the unique lipid profile of microorganism. And we have the proteomic analysis. In this, bacterial identification can be based on proteins produced under specific growth conditions with human body. This type of identification are called proteomic analysis. So in order to perform proteomic analysis, proteins from pathogen are first separated by high pressure liquid chromatography, and the collected fractions are then digested to use smaller peptide fragment. So that's the uh, technique. Then another one is proteins in the plasma membrane bind to lectins or antibodies, and this also can be used for identification. And how do you use this one? There is what is called this clumping together of the cells. Is a serological test, and we use it in grouping, in lance feed grouping. We talk about this lance feed grouping in when we taught streptococcus streptococcus species here, and they use it in the identification of streptococcus species. Then the second topic we are going to treat today is some fluorescent antibody methods. We have different types of fluorescent antibody method. We have the direct, the indirect, and the flow cytometry method for the fluorescent antibody method. This technique is very sensitive, and then they can identify a wide range of organisms and diseases in the laboratory. And then in fluorescent techniques, like I told you, they are very specific and sensitive. So there is what we call fluorescent antibody base, which require fluorescent microscope. Without fluorescent microscope, you cannot carry out fluorescent antibody method. And in this, a fluorescent substance absorbs light at one wavelength and emits light of a longer wavelength. So there is what we call fluorescein, which fluoresces an intense apple green color when excited under fluorescent microscopy. So this fluorescent antibody method is just a chemical manipulation in labeling antibody with fluorescent dyes to permit detection by direct microscopy examination. That's all about fluorescent antibody. Then let us discuss the three types that we have. In the direct technique, otherwise known as a direct immunofluorescence, a fluorescent antibody is used to detect antigen-antibody reaction at a microscopic level. This technique involves the use of single fluorescently labeled primary antibody that is used to bind a target antigen. So your target antigen is in your sample. It can come as a mycoplasma pneumonia, Legionella pneumophilia. So they use them to for rapid diagnosis of bacterial diseases. They are also used for streptococcus, the identification of Streptococcus pyogenes. So if the unknown antigen is Neisseria gonorrhea, the known antibody against Neisseria gonorrhea with attached fluorescent dye will bind to the bacterium and will not wash up. What this one is saying is that If your unknown antigen is Neisseria gonorrhea, you must have a known antibody that has been labeled with fluorescent dye, and this one will bind with that Neisseria gonorrhea, and it will not wash off. And once you view it under the microscope, it will fluoresce. So that's the example of direct uh, immunofluorescence. So we have indirect immunofluorescence, and this one is based on the ability of antibody to react with antigen as well as act as antigen and react with antibody. So that's why it's called indirect. Is the antibody will react with antigen. At the same time, the antibody will serve as antigen and react with another antibody, and this antibody is called anti-immunoglobulin. So it's a two-step process that begins with application of unlabeled primary antibody onto the sample that's directed against target antigen. So the second step of indirect immunofluorescence involves the use of fluorescent labeled secondary antibody that's directed against the FC portion of the primary antibody. You know, we have FAB portion and FC portion of any antibody. So this thing was taught in under immunoglobulin by are we there?
Yes, sir.
Yes, sir.
Or we are lost? Are we following?
We are following, sir.
Okay. So we this indirect is used in the detection of autoimmune antibodies. Example is SLE, that is systemic lupus erythematosus. So the example of the autoimmune antibody is SLE, systemic lupus erythematosus. So this technique is very essential in the detection of this kind of autoimmune antibodies. So the third one is the flow cytometry. And in the flow cytometry, it uses a fluorescent label antibody that can be used to quantify cells of a specific type in a complex mixture using flow cytometry. You can see the first two that we have discovered is more of qualitative detection, but this flow cytometry is quantitative. You want to know the number, and it's also an automated system and cell counting system that detects fluorescent cells as they pass through the narrow tube, one cell at a time. For example, in HIV infection, if the CD4 count is less than 500, that means that the patient is more likely to acquire opportunistic infection. And then if the CD4 count is less than 200, that means that the patient can no longer mount a useful adaptive immune response at all. So that means that anything that comes, it will take him off the ground. So this is that's the essence of using flow cytometry. If you want to know the number, if number is of essence to you, that is in case of HIV, then you will use flow cytometry. I think this is a convenient place for us to stop tonight, so that we will not bother ourselves. Tomorrow is another day. Let's stop here today, and then we can now ask our question.
Thank you, sir. Hello,
Thank
sir.
you. Thank you, madam. Yes, I'm hearing.
Please come again on catabolism and anabolism.
Okay.
Can you take up again?
Yes, when you talk about anabolism, is you want to form a complex molecule from smaller molecules. Anabolism, you are trying to build a very complex molecule from smaller molecules, so they will form a building blocks that will now become a real building by itself. It's like you want to build a house now. You will be setting the block one by one. At the end of the day, it will become a, a building. So that means that you start from the smaller one and becomes a complex. That's anabolism. Catabolism is that you are starting from complex to the smaller one. That means that if you have a building, you want to remove the roof, you want to remove the block, you want to remove the what's it called, the windows, you want to remove everything so that you will degrade the building so that it will now become smaller molecules. So that is the catabolism. And that's why you have catabolism of lipids, you have catabolism of carbohydrates. Why catabolism of carbohydrate is that you need energy to survive. We need glucose to survive. From carbohydrates, carbohydrates must be catabolized to form glucose. And then when there is no glucose around, the body must scavenge, or the body must find a way of finding glucose through another means. That is why there is catabolism of lipids and protein through the process of gluconeogenesis. Are we there?
Thank you, sir. Sir,
Yes.
all these methods like Krebs cycle and um,
Glycolysis.
gluconeogenesis,
Mm.
gluconeogenesis. So those are the methods that are being used in either catabolism or anabolism.
Yes, why
Am
all
I
these
right, sir?
methods? Yes, or why all these methods, all these I say terminologies are very important. Is that the microorganism needs all these things just like human being needs them before they can survive. We need energy for survival as human beings. So do microorganisms. So microorganisms also has to catabolize their energy so that their carbohydrates, so that it can be useful for them for their survival. Are you getting it? So
Yes, sir.
Yorùbá ní iṣẹ́ èèyàn ní iṣẹ́ ẹranko, bó ṣe ń ṣẹlẹ̀ sí àwa náà ló ń ṣẹlẹ̀ sí àwọn microorganism yẹn. That's the way it's happening to them as well. They cannot make use of that. They cannot make use of carbohydrate the way it is like that. It has to be catabolized into glucose that will be useful for their survival. Are we there?
Thank you, sir.
All right.
Sir, please, can you still come again on flow cytometry?
Yes, flow cytometry is just another method that is another method of what's it called? Fluorescent antibody technique or method. And what is flow cytometry seeks to do is an automated method where cell counting is of paramount importance. For example, for somebody that have HIV infection, the CD4, this CD4 count is very important. So this one, unlike the direct and indirect that are qualitative in nature, this one is quantitative in nature. You want to know the number, not that you want to know whether it is present or it is absent. For direct, what you just want to know is that there is what's it called? Neisseria gonorrhea, for example, or there is no Neisseria gonorrhea. For indirect, which detects autoimmune diseases or antibodies, example is SLE. systemic lupus erythematosus. So these ones also will tell us whether the autoimmune antibodies are there or not there. Are you getting it? But when you get to flow cytometry, it wants to know the number. And the number that are very important, the flow cytometry will give you the number. And any number that is below 500, it shows that the patient, the HIV patient, can acquire opportunistic infection if it is less than 500. But if it is less than 200, that means that the person cannot even mount any useful adaptive immune response at all. That means that anything that comes his way at that point in time, it will take him down. So, and that's why they use this flow cytometry, because we want to estimate the number of the CD4 T cells that is present in the HIV positive individual. Are we there?
Thank you, sir.
All right. Any other question? Any other question? Who was asking that question? Akinloye, Abi? Hello? Hello?
Yes, she was the one.
Okay. All right. Any other question?
No, sir.
No. Okay. Thank you for having me tonight. And then I think we are progressively coasting home, and then we'll still have another lecture tomorrow. So let's see, after tomorrow, let's see what next week will be like. But I believe that by tomorrow, I would have covered about 80% of the lectures that I'm supposed to deliver. So that means
Is
that...
it same time?
Hello?
Is it same time? The same time?
You say?
Is the lecture tomorrow lecture? Is it 8 p.m.?
It's 8 p.m. the same time.
Okay, sir.
The same time.
Noted, sir.
So
Noted,
that
sir.
let's see what we can do within the time that is available. And then it's better we take all our lectures now than to be rushing it. So that by the time we are doing revision, we will have read our notes at least once or twice. So whatever we are saying that time, it will be making sense during revision. So in the absence of no other question, can we call it a day?
Yes, sir.
Okay.
Good
Okay.
night, sir.
Good night, everybody.
Nice, sir.
Good night, sir. Good night, sir.
Thank you, sir.
Thank you. Let's thank God. We'll see tomorrow, God willing, and we'll discuss another topic of interest.
Okay, sir.
Dr.
Our
Ajayi,
notes, Oga.
you know my style now.
Yes, so
After the delivery, I will send a note immediately.
thank you, sir.
No problem. You're welcome. Dr. Ajayi, I greet you.
Yes, sir.
All right. Good night, everybody.
Good night,
Good
sir.
night, sir.
Hello, class rep. Hello, ma.
Hello.
Well done, my sister. Please, this other lecturer has not given us note on
Which
public
lecturer?
microbiology. Yes. Please, maybe you
On
should
which
help us to ask him. Hello?
on, on which topic?
On public microbiology, he has not given us public health. See me say more. So please help us to contact him.
When
Like
is
the
ready?
lecture we had today.
He's not ready
Yes?
with it yet. When it's ready, he will send it and I'll send it. He's not ready with it
Okay.
yet.
No, okay. No problem.
Thank
Thank
you.
you.
Yeah.
Okay. Okay. Bye.
Bye.
Bye.